Hello, everybody. Good evening. And nice to see all of you here. Welcome to UCR Palm Desert Center. My name is Agam Patel. I'm the executive director here. Pleasure to see all of you here for our third and final lecture in our three-part lecture series here with our friends at UCR's Division of Biomedical Sciences. We're so excited to have Dr. Monica Carson and Dr. Zheng here tonight to share some important research with all of you. Thank you all for joining us for this lecture series and many more here at UCR Palm Desert Center. Scott will mention a little bit about the survey that you saw outside, so be sure to complete that survey. But before we begin, just let me give you a few important reminders of events and programming that is coming up here at UCR Palm Desert Center. In just a couple of days, as part of our continuing lecture series called Wild Coachella Lecture Series, we are going to be joined by Danelle Baronia and Melanie Davis, researchers at the College of Natural and Agricultural Sciences. They are part of our Conservation Biology, Center for Conservation Biology, and they will be talking about the history of fire in the Mojave National Preserve. So that's going to be an interesting one here this week on Thursday at 6 p.m. On the 11th, as part of our ongoing collaboration with UCR's Jewish Studies program, we are going to be joined by Professor Rami Zwick. We are having our Fay Lecture in Jewry and Innovation on April 11th, featuring behavioral game theory. So join us for that next week. On the 24th, as part of our ongoing collaboration with the City of Palm Desert, our 16th year running the acclaimed public art documentary series, we're going to be featuring a film on Burning Man, the art, the sculptures, the stories, all that happens in Burning Man. This will be on April 24th. You won't want to miss that. That's going to be a special documentary. And then on May 1st, to close out our Arts and Letters lecture series, we're going to be joined by David Martinez, who's got a debut novel called Bones Worth Breaking. He's going to be in conversation with Todd Goldberg. So mark your calendars for all of those in April as well as in May, coming up right here at UCR Palm Desert Center. And I know I saw a number of partners here. Raise your hands, please, if you're a UCR Palm Desert Center partner. Thank you. I see a bunch of you out here. Thank you for your generosity and your ongoing support. We couldn't do this without you. An important reminder, folks, tomorrow and Thursday is our important UCR Give Day. If you haven't already gotten communication about that, you will shortly. It will help us continue our programming, our scholarships for local students, and much, much more. So please look for that communication and give if you can. All right, to help me welcome our featured speakers, please join me in welcoming Dr. Scott Pagan, who is Professor and Vice Chair of UCR's Division of Biomedical Sciences.
Thank you, everybody, for joining us tonight. As you see, and I think it was mentioned very eloquently, is that this is the third of our three-part series from the Division of Biomedical Sciences. And so we're happy that you join us today for brain and lifelong health of the brain. I have the pleasure today of introducing the chair of the Division of Biomedical Sciences. So she's my boss, so I'd really like, of course, a nice round of applause, please. My evaluation is coming up soon. And so when she's not, of course, leading the Division of Biomedical Sciences, she's also the Sue Johnson Presidential Endowed Chair for Glial-Neuronal Interactions, which also means that she's also the division, the center lead, the director of the Center of Glial-Neuronal Interactions. So with that, I will bring her up and introduce, let her have the stage. And then, of course, after that, we'll be accompanied by Sika Zheng, the director of the RNA Biology and Medicine Center. There you go.
Thank you so much for that introduction. And are we on? Okay, beautiful. It's looking at all these different things going on. Just so excited to be back here. Last time I was actually lecturing or just communicating and talking and engaging with people out here was pre-COVID. I've been part of the recent seminars that have been coming out here, but it's so exciting to be here, and I hope to be interacting with you. So today we're going to be talking about promoting lifelong brain health. And the whole point is, it's not all in your genes. So often we think, oh God, I got bad genes. Well, there you go. I might as well drink myself to death. You know, we don't want to go and do that. We have some control over our life. So we're going to talk about the role of genetics, life experiences, and chronic inflammation. And I'm going to be telling you stories from my lab, from my center, which is the Center for Glial-Neuronal Interactions. 20 years ago, when we were thinking about the name, I shouldn't have been so focused on being accurate. I should have called it what I would call it now, a Center for Lifelong Brain Health. So you get the idea. I do learn from things. And so we'll be telling you some stories, also snapshot stories from researchers in this group. Now, when we simplify this, what I'm really telling you is a story of healthy brain aging, or what a lot of folks are really worried about, age-associated dementia, and also other neurodegenerative disorders. But we're going to start with dementia because we all sort of want to remember who I is. You know, who am I? I'd like to know that. I'd like to have healthy aging. And so I'm going to be telling you a story of the experience you have along your journey. But we're going to talk about it from the perspective of this unusual cell, the microglia, call it the mom of the brain. And we're going to be talking about the brain's neuroinflammatory response. And lesson number one, your take-home message number one, is that context and perspective are everything. The same injury and insult is not the same for every part of the brain. So which context of the brain that was hurt, whether you're a genetic female or a genetic male, and also in the context of what kind of life choices you've done your whole life. I've had a life of really enjoying ice cream, and so that puts me in one context. There are other people who've done a lot of 5Ks and good, all these other things. That's a different context that these same events are happening in. And so lesson number two, I'm always giving you your take-home message. Yeah, it's really annoying. There's no magic little pill we can take that's going to just solve everything. It's really annoying. It's really true. What we always knew is sleep, exercise, staying connected. That is where you're going to make the most of whatever you were given and how you're going to be able to retain most of what you have and to keep developing it. Goodness, that means I have to take work and I can't stay up all night. So this is going to help us even in the face of toxic exposures and other kinds of illnesses that we will face. So when we're talking about this and we look at healthy aging, you know, we are always looking at this thing, you know, we go up to our peak and then it starts going down. We always think of that up and down. And when we look at this graph, we see like you get older and you don't want to end up in that red area, you know. As you get older, you want to slow down your aging, your biological aging. You want to bend the curve down. You don't want to be making life choices that are pushing you into the red. And so that's the challenge. How much do we control? How much is our genes? And what do we do? And let's say I already made bad choices. Now what do I do? How I want to make some good choices. So if we're thinking about that and we saw that little red area, what is that red area? Well, let's start about dementia. But we could also talk, if you're interested in the Q&A, some other diseases, some other neurodegenerative disorders that happen. But we're going to start with dementia. And mostly people are thinking, oh, how do I get it? Or better yet, how do I avoid it? But when we're talking about dementia, it's actually a bunch of different diseases. It's like saying, I don't want cancer. Yeah, I don't want cancer. I don't want any cancer. I don't want breast cancer. I don't want brain cancer. I don't want blood cancer. But those are all different cancers. There's different types of dementia. By and large, the biggest one, the risk factor with age, is Alzheimer's disease. Next is vascular dementia. But the rest of these come up as well. So that's what we're going to be talking about with dementia. But they're actually slightly different diseases. And each one of them, your brain and your logic is broken in different ways. So when we're thinking about dementia, what is it? Well, If you're like my grandmother, and she'd always call my grandfather, "You senile old fool, why are you doing that?" Well, mostly she was disagreeing with him. But if you really were senile, that is dementia, if you've really lost contact with things. But it's important to know that aging does not mean you become senile. Senility is a disease that is unhealthy aging. Okay? So it's a disease. It's not just a logical outcome to being older. Aging is one of the greatest risk factors for dementia, but we've also lived a lot of things. We may have made some bad choices. But as we age, do we change? Yes. Now, some people are going to say, "Oh, well, that's really depressing." It's not. It's a change-up. So, do we have an increase in forgetfulness as we get over 50? Yeah. It's not like at 50, but by 50, you can now measure self to self. I'm over 50. So if we think about my ability to remember now versus in my 20s, self-to-self comparison, I'm not as good. So am I depressed about that? No. I got a lot more to remember. I got a lot more in my head. I've got a bigger photo album in my head. Lots more going on. I just have a lot more interactions. So, okay, that's just happens. The other thing happens is slowing of response time. I'm not as fast to learn. I'm not as fast to recall. Just speed accuracy trade-offs. Not as fast, not as accurate. I'm just trying to grab those things out of my head. But I'm way better at pattern recognition. As we get older, there's the truth. Wisdom comes with age. And the race belongs to the swift and the young. Because I can tell my earlier self, yeah, run out in the parking lot. It's 80-some degrees. Go do that really quickly. At this age, I'd say, excuse me, why am I running out there? You know, we recognize patterns. When you compare self to self, as you get older, you have better pattern recognition. A lot of your neuronal networks are more complex. They're connected more. Pattern recognition. So if you're recognizing more, you can't be as fast on certain things. It's a trade-off. I give another example of that. My son, when he was in high school, had to make the choice. Do you want to be a swimmer or a runner? He liked both. But those muscle groups fight. You have to choose to be a runner or a swimmer. Our daughter, she liked music. She played clarinet and French horn. The embouchures fight. So her French horn teacher, which eventually became her primary thing, would always say, "You've been seeing the clarinet." Okay? So the same thing. We are different. That's okay. Let's embrace that. With wisdom, we can recognize the seasons. We can recognize the patterns of good, bad, and all sorts of things. So this is a good thing. It's good for our population, for the species survival, that we have both the swift and the wise. But when we think about dementia, it's a real thing. And dementia is severe loss of performance. And it has to be a loss. It can't be saying, "I can't play piano." Did you ever play piano? No. You had to have lost something first. So compare self to self. And so you start looking like, oh my goodness, I'm forgetting somebody's word. It's like, Scott, who were we talking with yesterday? You know, that will happen more and more. Okay. But I'll remember it later, and I basically know I would. It's very different where I've really lost myself, and I really can't find it. Similarly, yeah, we forget appointments from time to time. That will increase. But it's very different when you forget and you really can't operate. You actually need assistance to get through the day. So we're really looking at magnitude. Loss of what's called executive function. And when we look at some of these other things, yeah, I might forget what day it is occasionally, but I'll figure it out later. Versus I really don't know where I am, the season, what's going on. It's fine to get irritated. I like my coffee in the morning. I like watching a little sci-fi show at night, and I like an orange in the end of the day. If I don't get those, I can get a little grumpy. That's okay. I have a routine. That's very different than getting extremely agitated by having a slight disruption in your normal patterns. So that's what we're talking about when we're talking about, I like ways certain things versus doing the dementia. Similarly, when we're looking at losing things, getting tired, we're really looking at getting lost in some of these other areas as dementia. And another one to always think about, you might have changes of vision and hearing as you get older. Yes, you do. The physical apparatus is wearing out. But the problem is you get it corrected, and I still can't figure out this thing which doesn't have a good color, and what is it? I can't interpret what it is. I can see it, but I can't interpret it. So that's dementia, when you can't make sense of your sensory input. So that's where we're going with dementia. So when we think about this, we really want to say, how do I get there? And you might think, oh my goodness, I have bad genes. Everybody in my family gets this. It's in my genes. Not necessarily. Usually, you come from a family, you come from a group, you've lived in the same area, had the same exposures, you have the same exercise habits, the same food habits, and whatnot. So it could be common lifestyles with some common genes. And when we're thinking about what's going on beyond the genes, we're going to focus on the brain and the immune cell interaction. And one thing I always tell people to do is try to take one of your hairs and just hold it. Just reach your hairs right now. Just try to separate one of them. Every neuron in your brain is that far away from a blood vessel. Your brain is 2% of your body weight, yet takes 20% of your blood and 20% of the oxygen. So it's highly dependent. So guess that means heart health really is brain health. The other thing about the immune system is the immune system actually changes our behavior. And so if you get sick, you have a pathogen, you have an illness, it triggers sickness. So I have an inflammation outside my body. There's these things called cytokines to get into the blood. There's also some neuronal communications. And we show with an animal that will just lay down and, not get up. And it's because it's lethargic. It's going to have decreased social behavior. It's not going to have sexual interest. It's not going to want to explore. And that's a survival mechanism. If you're a herd animal, you're not going to infect your near kins. And if you're another kind of hierarchical animals, you're going to hide out so you're not taken down. So this is normal behavior. The problem is where the inflammation continues and drives this behavior too far. But it shows you the interactions. You do need the immune system interacting because without it, you have infections that will take over your brain and melt your brain down. There's a whole lot of things that happen when you're immune compromised that you have to protect yourself from. And the immune system is required to help you do memories and to learn things. So it's like in Goldilocks. Not too much, not too little. Let's just right. So when we think about dementia, there's all these annoying features that, from cardiovascular health, medicines that are required for our health as we age, chronic inflammatory disorders, HIV, neuro, AIDS, diabetes, diet, substance abuse, environment. These are all risk factors for different forms of dementia, specifically Alzheimer's disease. What do they have in common? Chronic inflammation. So we're altering that immune-brain interaction, and every neuron is within that close to the blood and the molecules that come through. So it's a dynamic interaction. And when we look at microglia, and we look at the brain, we see there are these cells called microglia. They're micro, little, they're glia, they're not neurons, and they're the little green things that are pretty there. And they're throughout the brain. And so I show you a low picture and a little bit of a close-up, and then I show you a closer-up one. And here I'm showing you, these are all the little microglia. You see them a little bit bigger, and then you see them bigger here. But there's also one right there. This microglia is hugging a blood vessel. This one is branched out and checking out what's going on with the neurons. By watching the microglia, we can learn a lot about the health of the brain because the microglia, they're moms of the brain. They're checking on the health of the brain. They are, just like that, they're helping neurons make play dates, putting synapses together. They are keeping the brain clean. They're taking away amyloid and all the bad trash and metabolism. And they will raise alarm and get very excited and call in immune cells and other things when there's damage. So when mom's happy, the family's happy, because that means everything's going well. But when mom's not happy, no one's happy, because everybody's walking on eggshells when mom's unhappy. I say things as moms. Dads are just important, but I'm a mom, so I tell you from my perspective. So we could have done dads of the brain. So we have a situation that when mom's unhappy, we don't know if she's going to respond well to the insults that are going on in the brain. And in fact, here's an example of two pictures of a healthy brain. Here's green with microglia, just beautiful hands going out. They're checking, all those arms are checking on all the neurons, saying how they're doing. Whereas here's right after a concussion, and this is actually far from the hit, but the microglia are all angry. And now they are having very different functions. They're finding that there's a problem in the brain, and they change how the neurons interact with each other. They're going to actually change how our neural circuit works. So they're playing an important role. And here's another picture. When I'm looking at the microglia, what you're noticing is that here's the microglia in green. When it's happy, they help the neurons play well together. When they get alarmed, they might disrupt those neural connections because they think something's aberrant. They're actually trying to prevent the brain from getting overexcited and doing this thing called exocytotic cell death. So it actually starts off protective, but it can go too far. And indeed, when we look at injury, we can see up here, it's like a brain that just has purple and hardly any colors in it. That's an image of a healthy brain. Whereas when you have reactive microglia, you're seeing all this yellow and blue. Some of those pictures are 11 years after the brain injury. So microglia, just like mom, she can hold a grudge. She can remember. They remember. And this changes how they respond to things. And so when we think about that, there's other things that cause them to react. And so again, we have a picture here. And in the top one is two pictures from male mice, and the bottom is from female mice. And we can see that when you put them on what's called a obesity-inducing Western diet, the microglia go from being very happy to being crazy angry. Whereas in the females, nothing much happens. They were happy, they stay happy. So this is the context and the perspective. The microglia are saying that the female brain tolerates fat better than the male brain. We all speculate that's because, well, the sex that is genetically determined to bear the child has to be able to hoard fat and nutrients, whereas, no, the other ones don't. We also can see that there's this lung-brain connection. We know that there's communication, so that when there's inflammation in the lung, it affects the brain. And in fact, the brain has a direct connection of this neuron called the vagal nerve that connects to the brain stem that regulates how you breathe. And what we find is that when you expose and cause, like when we're using mouse models, when you expose and cause asthma and lung inflammation, this actually causes the microglia to go from happy to crazed in the females this time and not the males. And we find that this causes changes in the neurons in the females where they're taking away the play days. They're pulling the neurons apart. So again, systemic inflammation, this communication causes a change. We actually tested, was this just molecules diffusing up there, or did it require that vagal nerve? We did ways to ablate the nerve, and we found out it was actually through that nerve that connected the lung and the brain. We've done the same thing with aging, and we find that males and females respond differently to systemic inflammation, with females being more responsive earlier than males. And then finally, we can show that sleep makes a difference. With sleep, the microglia and the other glia actually, like a sponge, squeeze out the brain and help clear it from metabolic waste. So, told you a story about the moms of the brain. When they're happy, we're all happy. When they're angry, upset, is it because there's been a harm or they're overly activated? But they're going to be at a different set point to respond to future insults. And so this is the big challenge when we're doing therapies in the brain. We can use the microglia to monitor and have them tell us what's healthy and disease in the brain, and then we can try to manipulate them to actually help the brain get better. And so I end by telling you that microglia are long-lived, they remember trauma, and they can adapt well or poorly, and that can be for decades. The brain needs an intact vasculature. Neuronal activity helps moms stay happy. So if the neural activity is not good, they get upset. So we want to stay active, keep our brains active. And yes, we have genetics, but we have a lot that we can do to foster good brain health. So with that, I thank you for your time and attention.
Thanks, Monica, for sharing us and helping us try to keep out of that red zone in terms of brain health. And with that, of course, naturally, at the end of this, we'll have a question-and-answer session for Monica and our next speaker that I'm happy to introduce. He's a colleague of mine. He's also the director of the RNA Biology and Center for RNA Biology and Medicine, Sika Zheng. He joined us about 10 years ago. And we're happy to join him on his journey as a faculty member and all the wonderful, exciting discoveries that he has made. And some of those I'm glad he's going to be able to share with you this evening. So without further ado, I'll let Sika take on the stage. Let's give a round of applause for Sika.
Thank you, Scott. Thank you very much. Thank you all being here. And it's lots of pressure after our chair's talk. Thank you, Monica, for a wonderful talk. So today I'm going to briefly touch upon a little bit about the research in my lab, but also again talk about the new center, the Center for RNA Biology and Medicine at UCR, and hopefully that can stimulate some excitement here. Okay, so first of all, who want to live a long life? Raise your hands. Oh, some don't. Okay. Okay, but that's important. So who want to live a long and healthy life? There you go. Right. That's it. Universally. That's great. Sure. Yes, as Monica mentioned, it's not just in genes. It's lots of environment, everything. But the organism also, during evolution, building a system to allow you to live a long period of time. Right? So, again, thank you. Again, speak louder. It's never important enough to stress that we actually can live 100 years. And that's not just a possibility, but that's probably going to be the future for for humankind. And the important thing here is that can I answer your question or? Okay. Feel free to ask questions during my talk as well. Now, the important thing is that the big barrier for longevity is your cells will die, particularly in the nervous system. So people over 80 years old, by statistics, have 50% of developing some symptoms of AD, Alzheimer's diseases. Right? And ADs or other late neurodegenerative disease has a hallmarks of neuronal cell death, just like what is shown over here. Right? But nevertheless, neurons actually can live a very long period of time. Many other neurons survive, even in the AD patients. So intrinsically, a cells can live a long period of time, 100 years old. And if you think about that, that's amazing. Because for every researchers in lab who handle cells, they know that cells are actually very fragile. They die easily. If you don't pay attention, like if a new student coming in, if they're not careful enough, the cell can die the next day. So for a cell to live such a long period of time is amazing, but it's actually evolutionary in trend and engineered to do that. Simply because, first of all, the neurons require to maintain your circuitry, right? Just as Monica mentioned, if your neuron die and then the associated pathway or circuitry is broken, then the associated function will lost. Right? For example, you attend this conference, but tomorrow you may forget about it if the neuron associated with it is lost, right? Or you may not be able to remember your childhood or maybe what you ate yesterday, things like that, if a neuron die out. Secondly, our brain actually have very limited regeneration capacity. So if a neuron died, there's no backup. There's literally no backup. So you are really born with what you have. If you're born with 100 billion neurons, that's the maximum number you have. It's only going to decrease only. So most neurons are intrinsically built to live very, very long period of time. And this is very, very difficult because at the same time, the organism is built with other pathway to induce so-called programmed cell death. This is some genetic program to kill cells when cells are under stresses. And actually is very much needed for wound healing, for your immune system. When your cells are infected by a virus, those cells need to die. Otherwise, the virus is going to keep proliferating and propagate, right? So it's made much needed as well. So there's this balance between longevity of neurons with this programmed cell that both require for longevity. Now the question is how the organism can balance that, because cell death is ubiquitous and it's always triggered when there's sufficient stresses. And stress is common. Who can tell me that you feel stresses from time to time? Right? Nobody is stress-free, right? So it's very common to have stresses. Now, so the question is, how do we study that? And can we learn something from it? And this is exactly what we do in the lab. First of all, we want to understand the genetic regulatory programs that enable neuronal longevity. How a cell can live 100 years old. Can we learn something from it? Can we harness that built-in mechanism and maybe hijack that to promote neuronal resilience against injury, against toxic stimulus for them to hang on for a long period of time? If you get a hang on, the longer they hang on, the longer you live with a healthy cognition, behavior functions, right? And second things, we want to understand neuronal regulation of life-death decision in neurodegenerative diseases. Right? So we understand, first thing we want to know what's a natural way to live long. And the second thing we want to know, hey, what triggered the cell death? Why do they die at a certain period of time at a certain point, particularly in aging or in neurodegenerative diseases? So at UCR with fascinating facility supports, department, campus support facilities, as well as wonderful teams, our lab was able to make lots of strides in that front. List some of our publications here. And I'm going to highlight one study we've been doing. And in this study, as we said, I asked very simple questions. So how neuron can live a long period of time but other cells don't? Because in any given second, actually about 10,000 cells dying in your body, get replaced, and this turnover is absolutely required for homeostasis, for maintaining healthy cells for your skins, for your liver, for your kidneys and everything. But except your brain, because in the brain you don't want to replace the neuron. They need to stay there so that you can maintain your memories. Right? So what we've been studying is we compare your brain cells with other cell types to figure out what's difference in the brain compared to other cell types. Now, what's interesting is that all of these cells share the same genome. They have the same DNA blueprint. They all come from the germ cells or zygote cells, right? That inherited from your parents. So their genome is the same, the DNA is the same. But what's different? Their RNA is different, right? So we compare the RNA in neurons with non-neuron cells. We figure out what's different, and we focus on cells that can induce, focus on genes that can induce cell death that I mentioned about. And what we found is very interesting and illustrated here. In what's it called? Neural progenitor cell or neural stem cells, and actually also in all the non-neuron cells. You're going to produce this particular transcript that belongs to a so-called cell death gene or pro-apoptotic gene called BAC1. So this is a gene that's very important to induce cell death in general cells. It's so-called commitment step. When it's activated, you kill the cells. It's like a trigger. And these cells basically determine the cell death competence. So how ready the cell will commit suicide under stresses. The more it has, the cells is more ready to die off under stresses. They're very fragile in some way, right? So in non-neural cell, this protein is highly expressed from its transcripts. But what we found is that when neurons started to differentiate, so they're coming from neural stem cell, and when they differentiate, they started to produce a different transcript from the same gene. It's still the same gene, it's still the background genes. But its transcript started to differ in some way that it included this particular small axon we call microaxon because it's only 20 nucleotide. And this 20 nucleotide, once inserted in the final mRNA transcript, is sufficient to kill out the protein. The protein is never made, actually, because the transcript is get degraded instead of being generated, funneled through a translational machinery to create the proteins. As a result, the protein is gone in neurons. So neuron can no longer commit suicide, actually, even under certain stresses for this particular cell death pathway. So they become very resilient, or should I say they actually can proactively evade cell death even under certain stresses. They just hang on. They just like, hey, no, whatever stresses that kill your skin cells, I don't care. I just want to hang on, and I want to do my stuff. And they do. So why do we know that it's important? Well, just like everything we do in the lab, we take it out and see what happened, right? So we knock out this axon, we take it out using CRISPR, we force a neuron to create this transcript that's usually only used by non-neuronal cells, because neurons usually create this transcript. And so we force neuron to retain the protein back. Now neurons start to use this protein that they normally don't. And as a result, in animal models, they die within 4 days after birth. They cannot even live beyond a week. So this tells you how important this small piece of sequence is in controlling neuronal survival and organism survival. Right? So, a microaxon increases neuronal resilience as it required for survival. And this is obviously an important trigger for neurons to change its competence to commit suicide. And if we can block this trigger, then neuron will not commit suicide. Right? So right now we're studying whether this is a mechanism that maybe through environmental stresses or cell-cell interaction that triggers the neurons to turn its expression from this transcript to this transcript so that it starts to express protein that allow them to commit suicide. And if we can block that, that will eventually allow us to manipulate neuron nervous system to allow neuron to live much longer period of time. So as I said, all of this all happened just at the RNA level, right? The cells share the same genome across different cell types, neuron and neural cells share the same genome, but they differ only at RNA. And that's what RNA is very appealing. For those who may not know the central dogma, the DNA is used to produce this so-called intermediacy molecule called RNA. We used to consider just a not so important molecules, but this RNA is used as a template to produce protein, which widely recognized is a very important functional molecules inside a cell to do all the cool work. But now we started to realize RNAs play a very important role in regulating different things inside the cells to make the cells look different, to sense the environment, as well as to conduct some critical regulation to determine the protein functions. So why is this important? This is interesting because we're now entering an age where lots of RNA technology become available. So you may have heard of COVID vaccine, right? The mRNA vaccine, and this is only recently become news, but it actually been developed for many years and become very well heard in the news. But also, there's CRISPR. This is an RNA-guided RNA editing tools that allows you to change the genome. All these tools are going to be available. It's actually being used in industrialized manner in animals, but not in human yet. But we envision in the future it can be used in different ways. But most important thing is that for lots of drugs, they're mostly targeting proteins, right? Small molecules, antibodies, target protein. But remember, proteins are produced from RNA. So if you can target protein, you can also target upstream RNA molecules. And targeting RNA molecules is way easier than targeting proteins. Okay. Lots of proteins are actually very difficult to drug, the so-called undruggable protein. But now with RNA, it's way easier because there's a sequence. Specificity of RNA is very simple, 4 nucleotide, AUCG, instead of 20 amino acids in protein, which can fold in a very complicated structure. But RNA is so simple, 4 nucleotides. And you can identify, you can sequence your genome under $1,000, right, these days. So it can be very easy to identify your genomes, and you can target whatever protein you want, a gene you want. And talking about sequence, this brings out another level, the precision medicine or personalized medicine. Our genomes share 99%, but it's the 1%, or actually the 0.5% that make us different between each other, make us different susceptibility to different diseases, and sometimes the cause of diseases. And that's what we want to treat or want to target, the root cause for your diseases. And this, we're only through understanding your genome and also understand what the RNA is transcribed, we can target those susceptibility locus directly and potentially cure the diseases. And with these all new technologies, I'm listing here most recently FDA approved RNA therapeutics. Actually, it just started since 2016, but it's accelerating because it's so easy to design once you have a target, and it's so effective. It's going to have more. I'm just going to show a video for the first one, the new nusinersen. Excuse me. And this is a drug also treated for spinal rasa to treat spinal muscular atrophy. This is a disease, a catastrophic disease. If you have it, actually is basically death sentences. Mostly for severe cases, is within 3 years for children. And most kids will not be able to live beyond 10 years old. But now with this, they can live more or less a normal life. So I'm going to show a very short video to tell you Emma's story, who is the first patient receiving this drug. And she's going to explain to you what it is and how it works. Way better than I do. That's why I show it.
Hi, I'm Emma, and I have spinal muscular atrophy, or SMA. Spinraza is a treatment you may be considering. Today, I'm going to tell you how Spinraza gets inside of the body and what it does once it's in there. You may hear your doctor talk about a procedure called a lumbar puncture or an intrathecal injection and are wondering, what is that? Well, it's a type of injection. And even though injections aren't anyone's favorite, it's the way to get medicine like Spinraza into the body. Spinraza needs to get to the central nervous system, and it uses the fluid in your spine to get there. The spine acts like a tunnel for messages to travel between your brain and the rest of your body. For people with SMA, certain messages have trouble being delivered or understood. Under the guidance of an experienced doctor, Spinraza gets injected into the back, goes up the spine and directly into the CNS. Once Spinraza gets into the body, it works to get those messages traveling properly. To understand how Spinraza does this, we need to talk about genes. Genes are inside of everyone, and they contain all the important stuff that makes you, you. You get your genes from your parents. I got my brown hair from my mom. There are a lot of different genes in our bodies. Some genes are in charge of eye color. Some are in charge of how tall you are. And there's another gene that helps your body talk to your muscles. The name of that gene is SMN1, and it makes a protein that motor neurons need to function. Motor neurons are these nerve cells that control how your muscles move. Say you're trying to move your finger. You think, I want to move my finger. Your brain takes that thought and tries to make your hand move by sending the message to your finger through a motor neuron. But for people with SMA, the SMN1 gene is either missing or mutated and can't make the protein for your motor neurons to send the messages to your muscles. If your brain can't talk to your muscles the way it should, your muscles can get weaker until sometimes you can't even move them anymore. But here's the thing. The SMN1 gene has a copy called SMN2. The SMN2 gene does the same thing as its copy, but it's only strong enough to make a little tiny bit of the protein that motor neurons need. This is where Spinraza comes in. The SMN2 gene has 9 parts. These are called exons. Imagine the SMN2 gene is a recipe, but the 7th step is missing. Without it, the food doesn't taste good. Spinraza works with the SMN2 gene to add the missing 7th step, which gives people with SMA more of the protein their motor neurons need to give their muscles a chance at moving. Now that we've talked about how Spinraza works, here is some safety information you should think about if you are considering treatment. If you have any of your own questions, be sure to ask your doctor. One of the things my doctor...
I'm not going to bore you with all the fine print for the drugs. But you can see how powerful it is, right? How this kid, Emma is an amazing story that she would not be actually living without these RNA drugs. So with that, we decided to build an RNA center at UC Riverside, recognizing the strength of the campus already have. But our faculty actually spread out across campus in different departments. They have very strong research for their own their frontiers. And so the center encompasses 3 different colleges, have 36 investigators in 7 or 8 departments as we're increasing numbers as well. This is really a multidisciplinary research center where we bring brain and minds together, try to stimulate something that would not be possible without the center. So we're going to focus on unraveling the secret RNA and facilitate this kind of interdisciplinary interactions for collaboration and train next generation scientists. This is the inaugural symposium we've done. Actually, this gentleman here, the keynote speaker, is a person who discovered the Spinraza drug. And for that, he was awarded a breakthrough prize in life sciences in 2019. A second symposium, this is last year. Then we attracted very good speakers across nation. And the keynote speaker here, this is another gentleman, Dr. Don Cleveland. He's right now doing the similar RNA testing drugs on many neurodegenerative diseases. So with all of this, I just want to leave you some take-home message here. The RNA is really, really important. This is a focal point of research in RNA Center in my lab, as well as many other labs. We want to unveil the language of life, right? How RNA can encode the blueprint of your DNA for a healthier, more resilient lifespan. And this is actually emerging as a new technology for healthcare as well as biotechnological breakthroughs. So with that, we need your support, which can be our catalyst to our success in research. Thank you.
There we go. Okay, that sounds a lot better. Sounds better to everybody else? Good, awesome. So basically this is our third series. As was mentioned, we have a survey at the back if you haven't filled that out already, because we're interested in knowing what you would like to hear from the School of Medicine. Also with that, we also want to provide these guys with some excellent questions. So who's first? Okay. I think we have one up there first, then we have a gentleman down here. Oh, over there first? We're excellent.
Hello. Hi. Thank you so much. That was absolutely fascinating. I have two questions, one for each of you, and I apologize I'm not addressing you by name. But the first question has to do with hyperbaric chamber, oxygen therapy, and those who have undergone a traumatic brain injury. And the second question is RNA and infertility, specifically with third-party egg donation and the ability to use CRISPR and as it's been used and the expansion of that in the infertility arena.
I guess I'd like to know a little clarification. What is your question about the hyperbaric?
I guess my question is, again, the internet, Google, blame Google, that it's been used successfully with people in terms of regeneration, successfully with some people, some individuals, obviously this is a generalization, with traumatic brain injury through a concussion as well as stroke. And I wanted to see what the research is from your perspective and your thoughts on it.
It's a really interesting question. That's not an area that I have high expertise in, but it's like a variety of things where you're manipulating the oxygen, basically because you're really changing that blood immune communication, that load. And so people have used in a lot of different directions, manipulating it to see whether you would provoke the brain to try to recognize injury and do a repair. So you're sort of yelling at it again and saying, do something. Right now, the studies are all over the place, to be very honest. It's going to be very context specific. Genetic males and females, and also where you are hormonally and all that affects this. It affects what kind of traumatic brain injury you had. And so it's a very complex thing. It's like saying, I'm going to have radiation therapy for cancer. Well, which one? Am I going to have surgery? So there's a lot there, but we need a lot more to be able to explore it. But with the other question.
Can I also ask a clarification? So are you asking whether CRISPR is being used to treat infertility?
Well, actually, let me clarify. RNA therapies for infertility more specifically.
I think that's a good question.
Apologies. I get my CRISPR and my RNA mixed up.
Yeah, that's okay. Yeah, so as far as I know, I'm not aware of anything like that. So I don't really have a good answer for you for that. Yeah. I'm not aware of ongoing clinical trials for treating infertility using RNA either. Yeah.
Great, thanks. I think we're moving on to the next.
I also have two questions. I believe RNA manipulation, or whatever the correct term, was used to a reverse sickle cell in a patient, I think on the East Coast. Has it been used in any other genetic diseases like Tay-Sachs or anything like that? That's the first part. Go ahead.
Yeah. So, you are right. So sickle cell is the first cases that people try to tackle, particularly with CRISPR, not only in the US, but also as a success case in Europe as well. So whether that's going to be broadly applicable is not clear, but the efficacy is amazing. So it's most likely going to work, in my own opinion. And I think we just need maybe a little bit bigger clinical trials to get approval. Yeah.
And the second question, I'm very confused about the dementia list. What do amyloid proteins, is it too much protein and that's what makes, and so the body is overproducing the protein and you need something to stop it? Or what is the connection?
You can think of amyloid as like a bruise. There's a bruise there. Am I worried that I have blood thinners on bruising, or he's hitting me all the time? And so amyloid is a sign that there's a problem. And clearly having the amyloid pile up is an indication there's a problem. We know that if you have a lot of brain trauma, like you hit your head a lot, we also know if you go under sleep deprivation, we know if you do other kinds of injuries to your brain, amyloid accumulates. And there's a correlation between more amyloid and you having really poor cognitive function. But there have been so many therapies designed at cleaning up and just sort of like vacuuming up the amyloid. And they've been a little helpful, but pretty marginal. You know, you would think if it was that bad, that should be caused, which makes everybody think it's like the bruise. He's actually hitting me. I'm sorry, not really, but you know, I mean that I should be saying this is an indication, but what else is going on? And so that's where a lot of therapies are. And that's why you start hearing this other thing called tau, and we hear some other kinds of issues. But amyloid piling up isn't a good thing.
Thanks. And I think we have one more, and then we'll get to the gentleman in the front.
Good evening. My question has to do with our diets. Does that play into play here? And also the inflammation. I understand there's certain foods to help us reduce inflammation in our bodies. And I was once told by a doctor here in Palm Springs that there's 4 indicators through a blood test that he could tell whether or not I was going to have dementia. He said there's 4, and he said I had 2 of them, and I don't know how true this is. Thank you.
So let's just take them in order. Diet, yes. Yeah, we should try to stay moderate in all these things. But if you had to put it in order, heart health is brain health. Number 1, keep your vascular health. Attack these problems as you can get at them. In an ideal world, you'll do all of these things, but start somewhere. So you need exercise, you need that blood flowing because that brain's using 20% of your blood volume. It needs that much oxygen going in there just to keep it going and alive. Diet, yes. When you have a lot of fat accumulate, especially as you age, the fat actually causes inflammation. Not all kinds of fat, like there's this thing called brown fat, that's a good kind of fat, but the fat that we're getting from eating all my favorite ice creams and all the things I like to eat, that's not doing me any favors. I'm getting low-grade inflammation. It depends if you're male or female. Males are going to suffer, especially with brain inflammation, earlier than females. Females will eventually be, it's just a slightly different threshold. So diet is important. Indicators in the blood. I have no clue which 4 he was trying to sell you on and which she was doing. Are there indices in your blood that are suggesting that you have poor vascular health and are putting you at risk for vascular dementia? Yes. Are there things that are saying you have so much amyloid accumulating in your brain it's dumping out into the vasculature? Yes. Are there things called neurofilament, light chain and heavy chain dumping out of the blood? Yes. But they're not necessarily telling you that you have dementia in your brain. They're saying, you need to do some maintenance on your brain. You need to be paying attention to the system. We're starting to see a lot of junk up here. So there are starting now to be some blood tests that look like they will be accurate, but we're not quite there yet, but we should be in a few years.
Great. And then gentleman up here.
Thank you. Another great program. Thank you, both of you. A clarification, then a question, if I may. Was it neurons that you said were born with millions of them and you don't get any more? Okay.
You are born with more than millions. You have 100 billion.
100 billion, okay. Are you surprised then, given if we assume that the organism, us, wants to survive, it works hard to survive, that neurons would be such a weak link in a sense, that they are not self-generating. They sometimes don't have all the pieces in place in the right spot. Are you surprised by that it wouldn't be naturally regenerating to keep the organism alive?
Yeah, that's a very good question. I would. But once you read, maybe I'll teach neuroscience long enough, you start to recognize the power of evolution and biology of itself. And indeed, that's a very good question. So in lower organism species, neurons does get replenished. I mean, the brain has the ability to replenish neurons, like worms. And sometimes they can also live with some of the neurons just die out completely, no problem. Even the animal model like mouse we have in the lab, they can live without a full brain. They keep going on. They just have normal lifespan. But I guess just as in higher organisms or like humans, you become so much dependent on so many different things. You're doing more and more very sophisticated stuff, and the function become more and more specialized in some way. And neuron not necessarily built to conduct a specific functions. Your whole body could coordinate and the different tissues coordinate to achieve specific functions. So it's not just the neurons work. In that sense, I think the nervous system is built on as a command to make things to work, but in the sense that for a species to survive, it doesn't necessarily rely on just one individual or one neuron to pass on the genes, right? So whatever can achieve the maximum fitness for the whole group is what helping that species to survive. And I think for us, for human, it's not necessary for individual to survive.
And I'd also like to point out, not only do you work hard to have certain neuronal connections that if you regenerated, you wouldn't get them again, so you would lose a memory, you'd lose a skill. We also have not common diseases, but we do have some diseases where you do have some aberrant regrowth. That has causes associated with seizures. That's commonly associated with a lot of dysregulation of your brain actually working. And so the brain actually not only has these long-lived ones to keep those memories and your skills there so that you are you, because your clone won't be you. It would have done something different. You also have other things, the glia, they'll make myelin, and they won't let things change because you worked hard to be you, and you want to have that you.
So promoting neuron growth isn't a positive way to address anything, is what I'm hearing. So, any other question? We have one there.
Hello, thank you. I think I know a fair amount about the brain in terms of it relates to Alzheimer's and dementia and learning and behavior, etc. But I've never heard about microglia. Microglia, sorry, until today. And I kind of wonder why. It seems rather important, and it doesn't seem to appear in the more popular literature or non-scientific medical literature. And then the second question is, how do you measure it? Can you do it through functional MRIs, or does it have to be done through autopsies?
Those TBI studies, those were imaging studies. I picked that one because it just had nice colors. It was easy to show at a distance. But there's a variety of PET imaging, some other kinds of imaging modalities where we can follow them. There's a whole lot of other things. Why you haven't heard of them when they are just delightful cells, and when you actually do studies like Dr. Zheng does and all these other things, a lot of the diseases of the brain map to dysfunction of the microglia. So you say, this should be a disease in the neuron, and you're saying, it affects the neuron, but it's the microglia not doing things properly. But they are little. For a century, they were really tough to study and to see, and neurons are big and they're clearly important, and we start with the lights brighter because we needed to. Yeah, that's true. So when you see those pictures on the TV or things, and they'll show you with this neuron, and it goes boop, 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 and it goes to thing, and it's like this neuron here, that neuron there, and a neuron over there, and there's space in between. Your brain is like beachfront property. There's no space between the cells. It's crowded there. It's very spatially controlled, and the microglia are in there. But they were called glia, the glue, because people focused on the neurons. So it's neuronal bias.
May I just add that neuron was discovered about 70 years before microglia is discovered. So it's just because simply a time lag.
I think we'll have a couple of questions, and then, of course, we'll wrap it up, and the speakers will be here after the session to answer any questions.
You mentioned during your presentation that it's not the genes. Well, if you do a blood test and you come up with APOE4, isn't that part of the genes that you can't change? So it's going to have a potential effect that you may get Alzheimer's.
So you're very right. So as much as I would like to be able to competitively play basketball, I am genetically 60 inches tall. So I can do sports, I could do things, but there are some practical limitations if I was to do certain other things. APOE4 predisposes you not only for inflammation in the brain, but inflammation in the body. It is a pro-inflammatory component. So if you have that risk factor, you should be mindful of things that are going to exacerbate inflammation. You're more susceptible to inflammation. So it's not a definitive sentence. It's saying you have a tendency toward chronic inflammation, pro-inflammation. You should take more care of that versus somebody who has a different inclination and may have different susceptibilities. So they definitely, your genes do put odds in your favor of one thing versus the other. But the reason APOE4 stays in the population is because those people are able to defend against other pathogens better than others who don't. So, it's a balance.
Great. I think we have time for one more question. Maybe two. We'll get the person down here in the front.
Is there any research going on to either slow or stop memory loss?
Well, I think that's the job of many, many people in the neuroscience community trying to stop or slow memory loss or dementia. We make progresses, and we have hiccups and setbacks. But I think, lots of people are pursuing that final goal, ultimate goal. I want to eventually, hopefully, benefit from those research as well.
But there's nothing on the forefront.
It's amazing how much exercise and diet, I hate to say it, really do this because it's really annoying because I know what I like to eat and I like to watch TV. There are elements and various therapies that are pretty much anti-inflammatories, trying to lower chronic inflammation. There are other things that are targeting certain neurotransmitter system. It sort of depends how your memory got broke. Was it more for a vascular disorder? Was it more for a neuronal disorder? And again, I put things like memory as saying you have cancer. Which cancer do you have determines what kind of treatment you're going to have and what the likely progression is. So some problems with memory where we really have no clue about how others are starting to be a little bit more tractable.
Thanks. And I think we have one more question here in the front, and then I think we'll wrap it up for the speakers. And if anybody else has questions, those speakers will stick around for a few minutes to answer.
I'm not sure exactly how to put this. I understand about CRISPR, but I'm wondering, when she explained about the genes, and she said that the gene for eye color, et cetera, but she was missing number 7. Now, how is it that you actually are able to isolate the particular one you want, whether it's sickle cell anemia or something else, and then do this? Is it a case of you do it outside the body and then you develop a vaccine or something? How does this actually work?
Yeah, that's a wonderful question as well. So you're talking about actually not just a combination of gene therapy, CRISPR, but also how it's delivered for specificity and all kind of things that are surrounding it. It all comes down to, for specific cases, for specific diseases. In terms of design for gene targeting or gene therapy in general, I'm talking about gene therapy, including CRISPR and others, AAV virus, all of this. It's not that difficult, I wanted to say. I mean, if I can train a high school student well, I mean, if he's super all day, super, committed, they can design something like that. But it's just whether you're targeting the right target or not is a difficult one. Well, sickle cell has been brought up time because it's such a simple disease from my genetic point of view. Because you have this one mutation, you got a disease. If you don't have mutation, you don't have disease. It's so simple, right? But for many other kind of diseases, as Monica mentioned, is a spectrum, is a risk factors. It's not a simple single disease. It has multifactorial combination. You sometimes don't know which one to target directly, so specifically, right? Like AD, you have, so far we have identified close to hundreds of risk factors, right? The A beta or ApoE has been talked about the most, but there are many others out there. So which one to target? We need to know your genome first to know which one to target, and we need to know which one's really contributing to the AD development, which one is higher, contribute more or less than others. And those contribution or weight could vary between individuals too. So those require lots of effort to really understand first. Well, and once you understand that, the delivery involving the specificity, but do you deliver that to neurons? Do you deliver it to the microglia cells or blood cells? And those involving also, again, not just the gene, but also how the gene is expressed in different cell types, right? So a lot of genes are actually mutation occurring in all the cell type, but they have specific tissue-specific phenotype. Some come down as dementia, others as an immune cell system defect. So they all vary because they're expressed differently in different places and different times. So we need to understand that. But we can also harness that for specificity. Why? Because if you want to target, let's say, microglia cell, the microglia cell surface may express something that no other cell type express. So why not we harness that and then tag our medicine or therapeutics with a ligand that can recognize that receptor. So that only microglia cell uptake the drug inside it to do the magic. Or no other cell type will care because they don't even recognize that. So there are lots of ways to engineer, to recognize this kind of cell type specificity to do some tricks.
Great. I think that's a great question to wrap up on. And just a reminder that we have that survey. And then also, I believe that most of these are going to be on YouTube eventually, right?
Yes, all of the lectures are recorded and will end up on our YouTube page.
So if you missed something, you can catch it there. And, of course, their speakers will stick around for a few minutes to answer any questions they weren't able to get to in the general audience. And I'll hand it back over to our lovely, grateful host here.
Thank you. Thank you, Scott. And please join me in thanking Dr. Monica Carson, Dr. Zheng. We hope you enjoyed this lecture series and many more. Please don't forget to fill out the survey. We want to hear from you. We want to learn from you. And this will help us inform our next lecture series, which will be coming up starting this fall. Otherwise, we wish you a good evening, and we'll see you here right back on Thursday. Thank you.